Werner syndrome, WS, often referred to as adult progeria, is a rare genetic disorder characterized by accelerated aging, early onset of age-related diseases, and pronounced metabolic and cellular dysfunction. While WS is uncommon, research into this condition has provided remarkable insights into the potential of nutrient-based therapies, particularly vitamin B3 derivatives, to target the fundamental pathways of aging. Studies in WS serve as a model for understanding how interventions like nicotinamide riboside, NR, nicotinamide mononucleotide, NMN, and other B3 precursors can support cellular energy, DNA repair, and metabolic resilience, offering lessons that extend far beyond rare disease to the broader field of longevity science. At the core of Werner syndrome is a mutation in the WRN gene, which impairs DNA repair and destabilizes the genome. This leads to rapid accumulation of cellular damage, mitochondrial dysfunction, and systemic metabolic stress, ultimately manifesting as premature cardiovascular disease, diabetes, osteoporosis, and other age-related conditions. Researchers have observed that NAD levels, critical for energy production, DNA repair, and stress resistance, are markedly depleted in cells affected by WS. This has led to the hypothesis that replenishing NAD through vitamin B3 precursors could restore some of the cellular resilience lost due to the WRN mutation. Early in vitro and animal studies have confirmed that NR and NMN can improve mitochondrial function, enhance DNA repair pathways, and reduce markers of oxidative stress in WS models, effectively slowing the cellular aging processes that drive disease progression. What makes WS particularly illuminating is that it acts as a magnified lens on normal aging. The accelerated decline observed in WS cells mirrors, in a compressed time frame, the slow deterioration that occurs in healthy individuals over decades. By studying how vitamin B3 precursors impact WS cells, scientists gain insight into fundamental aging mechanisms, particularly the role of NAD-dependent enzymes like sirtuins and PARPs in maintaining genomic stability and metabolic efficiency. In WS models, supplementation with NR or NMN has been associated with increased NAD, activation of sirtuins, enhanced mitochondrial biogenesis, and improved cellular stress responses. These effects suggest that B3 therapies may not only address rare disease pathology, but also provide strategies to reinforce resilience in normal aging. Another area where WS research has informed nutrient therapy is inflammation and metabolic balance. WS cells and patients exhibit chronic low-grade inflammation, insulin resistance, and dysregulated lipid metabolism. NAD restoration via B3 supplementation helps mitigate these metabolic stresses by enhancing mitochondrial energy output, reducing oxidative stress, and improving insulin sensitivity. Pilot human studies and early case reports suggest that NR supplementation in WS may lead to measurable improvements in metabolic markers, demonstrating that nutrient interventions can have clinically relevant outcomes even in the context of severe genetic disorders. Safety and tolerability are particularly important when translating findings from rare diseases to broader populations. NR and NMN have shown strong safety profiles in both preclinical and clinical settings, with mild or no adverse effects reported in most trials. This suggests that nutrient-based approaches can provide targeted biological benefits without the systemic risks often associated with pharmaceuticals. Moreover, WS research emphasizes the importance of precision and timing. Intervening before irreversible damage accumulates appears to maximize the benefits of NAD restoration, an insight that may apply to aging and chronic disease management in the general population. The broader significance of WS research is the demonstration that nutrient therapy can directly modulate genetic and metabolic pathways associated with aging. It moves the concept of vitamins beyond prevention of deficiency, positioning them as active participants in cellular repair, mitochondrial optimization, and genomic maintenance. For clinicians and researchers, WS serves as a proof of concept that interventions like B3 supplementation can influence disease trajectory and potentially improve functional outcomes, offering a framework for translating these findings into therapies for more common age-related conditions. Ultimately, the study of Werner syndrome teaches a powerful lesson. Even in the face of profound genetic vulnerability, cellular resilience can be enhanced through carefully targeted nutritional strategies. Vitamin B3, by restoring NAD and supporting mitochondrial, metabolic, and genomic integrity, exemplifies how nutrient-based therapies can become central tools in combating accelerated and age-related decline. As research continues, WS may provide a roadmap for developing longevity-focused interventions that benefit not only rare disease patients, but also the broader population, pointing toward a future in which aging is actively managed, resilience is restored, and life can remain vibrant and productive for longer.